A very welcome to lecture number 3, where we are discussing about the microbiology series, and we have discussed in the lecture 2 about some historical aspects of microbiology. In this lecture, also we are going to learn something historical aspects related to timeline, what happened in the microbiological field. So the historical development in the field of microbiology. So from 2020s to 2015 Rosen Bacon, disease produced by living invisible living creatures. So Rosen Bacon has first time say that the disease which is produced by invisible living creatures. As we know, in 2022 there was no any microscope. Even people are not that much of aware if there is something that any minute creature is also available in the earth that can harm the disease or which can cause the disease. In 1546, Girolamo Fracastoro first time he discussed about that caused by Fracastoro disease was caused by minute seed or germ spread from person to person. So Fracastoro in 1546 said that disease was caused by minute seed or germ spread from person to person. In 1658, Kircher first recognized the significance of bacteria and other microbes in diseases. Again, 1665, Robert Hooke, who discovered the cell, he termed that what is cell. So he discovered cell in the cork bark tree. So that was dead cell, and in 1665 he used some simple microscope, not simple microscope. He used a very simple type of lenses, and he termed that was looking like a cell, and cell means like a room. So that the thing was look like a uh, room. So he told that he termed that the creature that is cell. Okay. Then Francisco Redi in 1668 demonstrated the fallacies in the spontaneous generation theory, what we have discussed in the earlier lecture. In 1676, Antony von Leeuwenhoek discovers animal cues, that is little animals, what he said on that time. 1688, why only Anton von Leeuwenhoek discovered? He discovered RBC, discovered sperm, a lot of things, because he was the only person on that time in whole world. He was the only person who was having microscope. So whatever he was getting, he was just checking everything. He was absorbing everything. So that's why Anton von Leeuwenhoek discovered many things. And the first times when he saw that tiny living creatures, he termed that tiny creatures as a animal kills. In 1688, Francisco Redi publishes work on spontaneous generation of maggots. In 1766, Lazzaro Spallanzani conducts experiment that dispute spontaneous generation. In 1786, Muller produced first classification of bacteria. In 1798, Edward Jenner introduces cowpox vaccination for smallpox. So on that time, smallpox was a very serious disease on that time, and there was no any medicine was available on that time in 1798. So Edward Jenner, he was a uh, doctor types. Like on that time we can't say that much of doctors, but uh, he was doing experiment. So there was one village where he found that the people who is suffering from cowpox, they are not getting affected by a smallpox. Okay, so he thought, why in whole country the people are suffering from a smallpox, but only that village was not suffering from smallpox. So he curious why it's happening. So he found that the people in dairy field who was living in that area, he found that people are living with they are also suffering from cowpox, but the cowpox disease was not that much of severe as compared to smallpox. So what he found that the people who is getting affected by cowpox, they are not getting any infection from the smallpox. So what he thought, he did one experiment. What he did, he just taken the he did inoculation. So what he did, he took the some cowpox, the person who affected by cowpox, okay, and he took that what we call it, that disease, and it transferred to the people who are suffering from smallpox. Actually, he found that if the cowpox, the person who was suffering from cowpox, they are not getting infected by smallpox. Then he found that this virus has like similar structure, not that much, but they belong from same category. So the people who are suffering from cowpox, they are not getting infected by smallpox. So what he did first time, who the person, because it was very difficult to cure the smallpox disease and it was very severe. People are getting affected, they are dying, and there was no medicine or vaccine available on that time. So what he did, the person who was healthy on that time, he transferred all these cowpox to the healthy person, and he found that when he transferred cowpox to the healthy person, that person is also not suffering from the smallpox. So that's how the first time vaccine has evolved. Vaccination is derived from Latin word that means vacina, and vacina means cow. So that this is the first vaccine was available, discovered by Edward Jenner and introduced by Edward Jenner. Okay, and he did experiment with a very young kid. Okay, so in 1799, Spallanzani attacks on the theory of spontaneous generation. In 1839, Swan and Sliding formulated the cell theory. Theodor Swan was a German zoologist and Matthias Sliding was a botanist. They both are formulated the theory of cell. Then 1857, Pasteur shows that lactic acid fermentation is due to a microorganisms. So Louis Pasteur, he developed the process of pasteurization. Okay, what you seeing in the milk nowadays, that the milk will be stored for a longer period without any damage or without any bacterial infection. So why it happens? Because of pasteurization. There are two types of pasteurization, as LT and HT. So we'll discuss this thing in the later series. So Rudolf Virchow, all cell originate from pre-existing cells. He told first time that Rudolf Virchow, he given some all cell originate from pre-existing cell. Then 1861, Pasteur shows that microorganisms do not arise by a spontaneous generation. So Pasteur shows by an experiment that all the microorganisms do not rise by a spontaneous generation. In 1866, Lister publishes his work on antiseptic surgery. Then 1869, John Mister discovers nucleic acid. 1876 and 77, Robert Koch demonstrated that anthrax is caused by Bacillus anthracis, that is a bacteria. So where we found Bacillus, that is a bacteria. Anthrax is a disease which is transferred from the person who was working in the uh, the cat many of the cattle are suffering from anthrax disease, anthrax bacteria cause. So many people in sheep, so people will just obtain the wool from the sheep, and they will send that wool to the uh, industry. So what many people found, the people who was just manufacturing the string of that wood, they are also get affected by anthrax. So this is the anthrax, and that was a bacteria which was going transferred from sheep to the even human beings. So that was a very serious disease. So first time Coach demonstrated that anthrax is caused by bacillus and anthracis. Then 1881, Coach cultures bacteria on gelatin. In 1882, Koch discovers tubercle bacillus. Then 1884, Koch postulates first published. Then in same time, the Metchnikoff describes of what is phagocytosis. Then autoclave was also developed in 1884. Then gram staining technique was also developed in 1884. Then in 1885, Pasteur developed rabies vaccine. I think everybody knows about rabies. Then 1887, Petri dish developed by Richard Petri. How Petri dish look like? It is spherical set. If you are if you are in college, if you ever visit laboratory, even in the school time, you will find the Petri dish that was discovered by Richard Petri. That's why this Petri plate name is Petri dish, because of his last name. They have He has given his name, so that's why he's Petri dish. Then 1892, Ivanovsky provides evidence for virus causation of tomato mosaic virus. Then 1897, Ross shows that malaria parasite is carried by the mosquito. Then 1899, Bejernik proves that a virus particle causes the tomato mosaic virus. Then in 1906, August Wassermann developed the first serologic test for syphilis. Syphilis is a sexual transmitted disease. Okay. Then 1908, Paul Ehrlich becomes the pioneer of modern chemotherapy to treat syphilis disease. Then in 1910, Francisco Ross discovers viruses that can induce cancer. In 1950 to 1970, F.D. Harrell and Todd independently discovered bacterial viruses. Then in 1923, first edition of Burgess manual. 1928, Griffith discovered bacterial transformation. 1929, Alexander Fleming discovers penicillin. Okay, so in 1929 he discovered penicillin, and from 1939 to 1945 World War II has been started. So many people are got saved because of penicillin. Because on that time before World War, the penicillin was also discovered. Then in 1935, Stanley crystallizes the DMB. In 1944, Avery shows that DNA carries information during transformation. Selman Waxman discovers streptomycin. In 1946, Lederberg and Tatum describe bacterial conjugation. In 1952, Hershey and Chase show that bacteriophage inject DNA into host cells. Zinder and Liederberg discover generalized transduction. In 1953, Watson and Crick propose the double helix structure for DNA. Then in 1954, Jonas Salk develops the first polio vaccine. In 1957, Isaacs and Lindman discovered the natural antiviral substance that is interferon. In 1958, Liederberg makes discoveries concerning genetic recombination and the organization of the genetic material of bacteria. In 1959, Korenberg and Okowa awarded Nobel Prize for the discovery of enzyme which produces artificial DNA and RNA. In 1966, Rouse discovered tumor-inducing viruses. Then in 1971, Theo Demer identifies viroids. In 1975, Kohler and Milstein developed technique for the production of monoclonal antibodies. In 1977, recognition of Archaeobacteria as distinct microbial group. Then in 1979, Henle identified first virus regularly associated with human cancer and insulin synthesized uric rDNA techniques. In 1982, recombinant hepatitis B vaccine developed. In 1982-83, Czech and Altman discovered catalytic RNA. In 1983 and 1984, Gallo and Mongenier isolated and identified HIV virus and PCR, that is polymerase chain reaction chain, developed by Mullis. 1990, first human gene therapy testing began. In 1995, Lewis, Nuslin and Wistens for the study of physiology of genetics of microbes. In 1997, Prussiner discovery of prions. So that is all about the historical aspects of microbiology what we have discussed today. Again we have to take one series about history. It will be little longer. I have divided in small lectures. So thank you very much for this